Welcome to this video on glucosidase inhibitors. Human glucosidase exists in two forms, alpha and beta. Alpha glucosidase is located in the small intestine and breaks down starch into glucose, whereas beta glucosidase is located in the liver and breaks down its corresponding substrate into glucose as well. The substrates themselves are classified as glycoconjugates, which means that essentially they are large units of carbohydrates. Both enzymes catalyze reactions by breaking down glycosidic bonds within their substrates, and essentially they go about a process called hydrolysis. Inhibitors of glucosidase are either naturally occurring or synthetically produced. The first natural inhibitor, called natriuretic, was discovered in the 1960s by Ishida et al. Not only was it an amino sugar, but it was also known to show antibiotic characteristics. And from this, other derivatives were formed from natriuretic. To name a few, you have 1-deoxynatriuretic and you have salicylic. Scientists thought, you know what, why not push it a little further? And other inhibitors were newly produced to be called transition state mimics. What do I mean by that? The idea was to create a molecule that was able to mimic the structure of the natural substrate in order to bind to the enzyme, block the active site, and prevent the enzyme from catalyzing its natural reaction. And hence, you're able to prevent diseases from occurring. And this proved to be really successful, as inhibitors such as arkebrose, miglatol, and voglibose was able to be produced, and these inhibitors are actually used in medicines today. Although these inhibitors are effective and are actually used in the drug market today, there have been some side effects. Patients who use these inhibitors experience symptoms of bloating, diarrhea, abdominal pain, and flatulence. Of course, this is highly undesirable, and industry is working really hard today to produce inhibitors that are not only high in their efficiency, low in toxicity, but show minimal side effects. The most successful leads can be broken down into four main categories: carbo sugars, amino sugars, thiosugars, and hybrids. Carbo sugars are able to mimic the transition state by having a carbon atom in replace for the endocyclic oxygen in the cyclic ring. A series of compounds 1 to 4 were produced by carbocyclization. Compounds 3 and 4 showed weaker inhibitory activity in comparison to compounds 1 and 2. This was thought to be due to the differences in the binding modes when these analogues bind to the active site in the actual enzyme. Amino sugars are able to mimic the transition state by having a nitrogen atom in replace for an endocyclic oxygen. Two compounds were analysed, top-DGAN and UV4. Top-DGAN is a derivative of 1-deoxynatriuretic, and it showed high selectivity for endoplasmic reticulum and is also a promising lead. UV4 is currently in stage 1 clinical trials to treat the dengue virus and is also known to be non-toxic. Thiosugars are classed as transition state mimics by having a sulfur atom in replace for an endocyclic oxygen or having a containing disulfur bridge. These compounds are thought to be able to treat disease. However, there has been some difficulties with the selectivity between the alpha and beta stereoisomers. Due to this reason, there has been little progression of this class of compound into clinical trials. Now coming on to hybrids, these analogues are a mixture of different compounds. Products show that analogues with combined chemical properties exhibit high potency against alpha-glucosidase. However, there have been little progression of this class of compounds into clinical trials. Out of the different inhibitor classes discussed, there have been large progression into treating three main diseases. Diabetes is a condition of extreme levels of glucose in the blood. Gaucher's disease is a lysosomal deficiency disorder. Viral diseases come under a very large bracket. However, in relation to glucosidase inhibitors, the main diseases include HIV, the dengue virus, the Zika virus, and influenza. Out of the different classes discussed, immunosugars have been the lead compounds to treat disease, and the lead analogue within this class is known to be UV4. It's currently in stage 1 clinical trials to treat the dengue virus. It's non-toxic, and it has the potential to treat the Zika virus. This highlights the diversity of this compound, as not only is it an alpha-glucosidase inhibitor, but it also shows much promise into treating other diseases in the future. Thanks for watching.